Okie. We're gonna start on with our next presentation. So Dr. Matthew Farrer is a professor of neurology at the University of Florida. So his research is sort of into genetics and neurological conditions, including Parkinson's. And so he's going to be talking today about the origin story of LRRK2 Parkinson's, from the what, where, and why to the how. So I will turn it over to Matt. Just turn on the lights.
Is it working? Is it working?
Yep, there you go.
Kind of working. Okay. So there's a lot to cover in 25 minutes. So I'm not sure I'm going to get through all of it. But I'm going to get through some of it. And any questions that you have, please ask, and I'll try and illuminate the best way I can. So I've been working in Parkinson's disease research for nearly 30 years, a long time. Came to North America when I was 27 and 57 now. And I started on this journey back then. I wanted to work on dementia, and I ended up at Mayo Clinic, going to work for John Hardy, thinking I'll learn a lot about statistical genetics, because that's what I was into at the time. I thought John would teach me everything I want to know about statistical genetics. And then I got there, everyone looked at me for the answers. But you're here to do that. And I'm like, I am, but I'm here to learn. And but thankfully he was a good mentor and sent me away to Cambridge and to Zurich and to different places to learn how to learn my craft. There wasn't enough room at Mayo to work on dementias, because there was a lot of big egos, big Jesus in the field. And I ended up working on Parkinson's disease. So I bumped into a guy called Manfred Munter, who is the chair of neurology in Mayo Scottsdale, and he's just retiring. And he said to me, Matt, I've had this enigma my entire career. It's his family, and they're from a little tiny podunk town called Kingsley, Iowa. And I've written this paper about them. The first patient from the family was seen at Mayo in 1924. And we've been trying to figure out what's their malady ever since. They have Parkinson's disease. And it's inherited. And you could clearly see that there was a dominant pattern of inheritance. By dominant, I mean that 50% of each generation gets it, on average, down the family line. It's passed from parents to their kids, generation after generation after generation. And this was an anathema at Mayo, because Parkinson's disease, and part of the reason I went there to study it, is known as a sporadic complex trait. There is no family history in Parkinson's disease back in the day. There is no genetics, doesn't exist, and there was a kind of considered to be a rather stupid endeavor to go into it in the first place. Indeed, I was told that by NIH. You're wasting your time. It's a stupid endeavor. There's no genetics. I will point out the heritability of Parkinson's disease is very low. Heritability is the amount of variability in the clinical expression or pathological expression of the disease. How much of that variance correlates with variability in the genome. And in PD, that's 27%. That's lower, far lower than Alzheimer's disease, far lower than most, pretty much all other neurodegenerative diseases. But nevertheless, it's important. And the reason it's important is because it's foundational. And why I say that is because we have a lot of associations, right, in this world. So many associations. I'll give you a good example. Birds have beaks. They're associated with beaks, right? Birds are associated with wings, flying. They're associated with feathers, right? Wonderful, wonderful associations. And you can make something of it. Okay, maybe the bird has these different features because it helps their lifestyle or whatever. And but does it help you conceive the egg? Think about that. Its etiology, its beginning, its start. Does those associations help you conceive the egg? The answer is no, it doesn't. So when I got into this field, the world was doing association studies, associations with paraquat and PTP. Well, PTP was pretty cool, but paraquat with well water, with Perhaps that's pesticide exposure with coffee drinking, smoking, lots and lots of associations. You've probably heard of many of them, right? But where do they lead you? Do they help you do they help me as a scientist, us, the world, understand what Parkinson's is and what's going wrong? And that's the reason for genetics, the type of genetics I do. So I call Mendelian genetics. It's not all the same. It's not all the same. Most people who study genetics in Parkinson's disease are looking at associations. And I think they're wonderful, but not useful. I'm going to show you, this is my concept of the disease. Very simple. It's a motor, right? Movement disorder, supposedly, movement initiation. It's far more than that. I mean, I look at Parkinson's disease like I look at an onion. It's got so many layers. There's the clinical layer and how people feel. And during the day and the week and the months and the years as they go by, and the temporal evolution of the disease. There's the physiologic aspect of it. Physiology with tissues and multiple cell types in those tissues, and how the organism responds, this parasympathetic, sympathetic kind of fight-flight issue in Parkinson's disease, which has gone awry. There's a metabolic piece to it. There's an inflammatory piece to it. It's all that's the physiologic layer, if you like, of the onion. Underneath all of that, there's a cellular and there's a molecular layer. And the molecular layer is driven by genetics, by genes. So getting to the heart of it is to understand that foundation. That's why we do it. Initially, we found synuclein. We saw this motor originally, clinical phenotype of Parkinson's disease. That's the analogy. But we didn't know the component parts. We don't even know how it's made. We don't know what it's comprised of. We don't know how to build a damn V engine, right? And if we don't know how to build it, how the hell are we going to fix it when it goes wrong? I don't know. So we found synuclein. We thought, oh, okay, maybe that's the, I don't know, piston cap or whatever. Got that bit. Found LRRK2. Found parking. Found pink one. And so it goes on. And all of those Mendelian genes now converge. All roads lead to Rome. They all converge on the same damn mechanism. And there's only one mechanism. And what it is, it's this. And it's evolutionary and it's ancient. It's basically, now I get to techno babble. It's lysosomal biogenesis around autophagosomes. That's partly where the story comes in. And what that is, it's basically the evolution of our genome in response to pathogens. And it happens over millennia. Didn't happen last week, happened over millennia. And our genome, to habit our ecological niche, has evolved over time. So has every living species on the planet. Their genomes evolve too. And there's this reciprocal fight, challenge between the two, because everything wants to survive. Everyone wants to, everything. The most humblest Salmonella typhi wants to survive at your cost, because it's going to cause gastroenteritis, but it wants to survive, right? And it's going to do its utmost to do that. And that might mean evolving its genome, because the ones that do survive are the ones that propagate next, the ones that spread, right? So a tiny tweak in the Salmonella genome helps its survival advantage. Same thing in the human host. Of course, it happens over a lot longer time intervals. Salmonella divide every 20 minutes. Human beings divide every 30 years, roughly, right? So it takes a lot longer for the human genome to change. But that's what's happened. And then these Mendelian gene mutations. So I'm going to talk about LRRK2, the what, the where, the why, the when, the how. But that's how LRRK2 mutations have come about as a response, an evolutionary response to pathogens. Nothing makes sense in biology unless it's in the light of evolution. I'm afraid it's very true. This guy, bloody brilliant, Ukrainian geneticist, and anyway, I won't go into it, but it's a fantastic read. And anyone can read it. It's not esoteric, it's about taxonomy and about the dawn of molecular biology and how all the taxonomists got it wrong, right? Prior to knowing what the DNA sequence was, prior to realizing that, hang on a minute, there's at least 7 species of giraffe, and they're not as closely related as you think they are. They're very different. The taxon was wrong. So I'm talking about the beginning for me was with my friend Jan. Jan was a great fisherman, when I say great fisherman, he took me fishing several times, and he did it in true Norwegian style. And that's not with a pole and a hook. No, no, no. What you do is rowing boat and a bloody great net, and you take it across the lake, you let down the net, and then you have to have a lot of patience. A lot of patience. Hours and hours, the whole day goes by. And at the end of the day, as the sun goes down, you go across the lake and you pull up that net, and up come all the fish. Right? Yeah, it was a great fish. And we went looking for families with Parkinson's disease in central and northern Norway. I told you I was working at Mayo. I met Jan at a meeting in Japan in 1998, I think it was. And he said, Matt, you must be doing so well. Mayo Clinic's wonderful. It's got all these resources, all these patients, all this material. You know, and I said to him, Jan, I was having a bad day. I said, yeah. I never met him before. Just having a cup of coffee. I said, yeah, no, they won't give me anything. They won't give me access to any patient material at Mayo. They told me I'm an idiot and I'm focusing genetics on a sporadic disease, wasting the time. So so he said, oh, but I've got lots and lots of patients. And he was the chair of neurology in Trondheim, central Norway. And I'll help you. Why not? I'll do the clinical stuff, you do the genetic stuff. And we had a wonderful relationship. Unfortunately, he died a few years ago. And but he recruited these families, and you can see they're quite interesting. Well, maybe you can. I can see them being quite interesting. They follow a dominant pattern of inheritance, so 50% each generation is affected, roughly. There are people that aren't affected. So this man here is deceased, okay, but he didn't get the disease. The disease is noted. Parkinson's disease is noted by a black symbol. And this lady here, for example, she's deceased too, but she didn't manifest the disease. It turns out they're all actually related to 15th century founder. Oh, if I get there. And they all have the LRRK2 G2019S mutation. When we discovered the mutation, we found it was here. It's the start of what's known as the activation segment, that bit in purple. So this ribbon of protein, a hole covers this cleft, this hole, if you like, where substrate comes in and becomes phosphorylated. We know what the substrate is now, it's called RABs, RAB GTPases. But they come into that pocket and become phosphorylated. And what's happened is this mutation, the glycine to serine, affects the hinge of this piece of ribbon of protein and keeps the doorway ajar. It's constantly open, right? So LRRK2 kinase is always constantly active. It's on the whole time. Shouldn't be on the whole time. It's meant to be on and off. So we think now, and probably the greatest hope for neuroprotection, I think, in Parkinson's disease, right now, and probably for many years to come, is LRRK2 kinase inhibitors. There's a lot of challenges. It's not going to be an easy road. Denali's got a huge responsibility here. They can't cook it up. but because there's a whole variety of inhibitors that will fit that pocket, block the doorway. Right? So it isn't always ajar. You won't necessarily have to be on it the whole time. It's just enough to stop it. I see Parkinson's disease as a vicious cycle. You know, it basically gets started and becomes self-perpetuating. If you can stop that vicious cycle, you can halt its progression, you can stop it. These families are all related. Here they are, 1, 2, 3, 4, 5, 6, 7. These are what's known as haplotypes. If you look around the length of chromosome, hypothetically speaking, my arm, the mutations in the middle, they all have the same variability. They're all cousins of each other. And that turned out to be the case. Not surprising, really. There's boats that go up and down the coast delivering the mail, and the postman, was he delivering something else? And these families, they're all related. But we found families in the States and Ireland and Poland. So this is where the where comes in. I won't get there yet. Anyway, doesn't matter. This is all about LRRK2. This is where the where comes in. At the time, I had a Spanish graduate student in my lecture, Marta, and he brought some samples for Parkinson's disease patients from northern Spain. And I also was working a little bit with Eduardo Tolosa in southern Spain. And when we found LRRK2 G2019S, I was thinking, my goodness, okay, it's weird. It's in Poland. It's Polish samples. It's in US samples, maybe. Okay, Norwegians go there. Vikings, you know, they got up to a lot of trouble. They went everywhere. But as we looked in the Spanish samples, we found that LRRK2, the frequency in Norway was 2.3%. The frequency of this mutation in northern Spain was 5%. As you go down southern Spain, the frequency gets higher. And I was very, very fortunate to get introduced to a guy called Lev Middleton, who ran the first Neuroscience Institute in Cyprus. I met him at Darwin's private club, the Athenaeum in London, many, many, many years ago. And I had dinner with him. It was really rare coincidence where Levkos had a window into the whole of the Mediterranean basin. He was an epidemiologist and a neurologist. And he said to me, Matt, do you know about Parkinson's disease in North Africa? I had no idea. Look in any medical textbook, says nothing. It's an epidemic. LRRK2 2019S was found in 1 in 2,000 people across the entire Maghreb. That's all the way from Morocco to Libya. Right? That's a population of Berbers of around 36 million people. That's probably something in the order of 20,000 patients with Parkinson's disease with LRRK2 2019S. So I thought, right, the mountain's got to go to Muhammad, right? So I did. This is Faisal Hantati, a good friend of mine and a wonderful collaborator for 20 plus years. And Rachel Gibson, I don't know who that guy is, he's just in the Rachel Gibson was at GSK at the time. This is 2005. And I said to Faisal, we didn't know about LRRK2 2019S. How about we develop a collaboration? We look in your patient populations at genetics of Parkinson's disease, because I realized that it was pandemic. So many Parkinson's patients. Social medicine in Tunis. And if you can't get treated or seen anywhere else, you can come to Tunis and you can get treated and get levodopa. And they will see you for Parkinson's disease. And it is overrun with patients, as you can imagine. That's an institute for a whole variety of things, MS, ALS, Parkinson's. And we started this collaboration. I brought the whole board of the Fox Foundation over there to try and get them to invest. And they did, to start, a little bit. Things went pear-shaped with the Arab Spring in 2010. Anyway, LRRK2 2019S, they have the same haplotype down here as the Norwegians do, as does everybody. 99% of people have a LRRK2 2019S mutation, are related. They're cousins. I said this to Sergey Brin. Sergey Brin has the LRRK2 2019S mutation, the head of Google. And he said they're going to have a big party. Google's campus is going to buy everybody come along. And I'm holding him to it. And if this is video on YouTube, Sergey, that's one for you. It's going to be expensive. We need a big party if we can. So everyone has the same mutation, and it's been driven. This hasn't. Normally, when mutations happen in the genome, they happen once, right, and they die out when that person dies. This mutation hasn't. It's increased in frequency over generations and generations. It's increased. And the reason why is it provides a positive selective advantage, a survival advantage to reproductive age for those people who have it. Pretty remarkable. You can see that as well, and I'll show you pictures of that. But there's some enigma about it. This is a family, a Tunisian family, and I went to see so many Tunisian families. Such a privilege to do that, to go into people's homes and to say, you know, help us out, tell us about the disease and tell us about what's going on, and to learn, because it's not in a medical textbook. Give you an example. I went to this lady's house. She was 53 years of age. She got the disease in her 40s. She had young onset Parkinson's disease, and she had 4 kids in the family. And I went with a couple of neurologists, and one of them, a friend of mine, John Duda, who's really good at olfaction, looking at sniffing sticks. And we talked to the kids, 4 kids in the family, and they were laughing and joking because they said, "Mum's always burning the cooking. She's always burning the cooking, you know, because there's always a fire every day over the stove, and everything's always burnt." And John says, "How come you don't smell the cooking? How come you don't smell it burning?" Goes out his sniffing sticks, and all the kids, there's 4 kids, I think their ages from 24 to like 36 years of age. Not one of them could smell the sniffing sticks. They all had hyposmia. We got them to walk outside, right? Just down the corridor. And this is how they walk. Do you notice anything unusual? I'm asymmetric. People should swing both of them. And if they don't, it's subtle, so subtle. You can feel it, that rigidity in the limbs. and I could feel it. And the 24-year-old, no movement disorder, no diagnosis. And then you start to ask them about things, sleep, constipation or gut discomfort, things that you start to pick it up. You start to understand that this disease happened a lot earlier. It isn't just a movement disorder, it's far more than that. It's an evolution of those symptoms. It's a vicious cycle. Anyway, from a movement disorder point of view, these are classified with UPDRS, Parkinson's disease. These boys, 40 years of age at onset, their mum and dad, their mum, they're both LRRK2 G2019S mutation carriers. Dad, when this was done, was 78, mum was 72, not affected. How carefully have they been examined for those very subtle features? I can't tell you, not very carefully. But obviously they don't meet criteria for a clinical diagnosis of Parkinson's disease. Far from it, right? So LRRK2 is not fully penetrant. The weird thing about it is it's population specific. This is a curve of LRRK2 G2019S patients with age of initial symptom onset on the bottom and cumulative incidence, how many people actually have manifested Parkinson's disease according to movement disorders specialists. And in Norwegians, in yellow here, you can see the shift. The curve is a lot later. Oh, and I said to Jan, maybe it's the neuroprotective advantage of eating a lot of salmon. You're not getting as much salmon in the Jewish population or in the Tunisian population, because their shift is 20 years younger. That's a big difference, you know, in terms of manifesting disease, in terms of the prevalence of the disease in the population. You know, by the age of 60 years of age, half of the G2019S carriers, more than that, are affected in Tunisia or in Israel, whereas in Norway, it's only a small number, maybe far less. Why is that? You want to know why? I'll show you why. Before I get there, the slides aren't dilly anywhere. I don't want to go over the slides. I want to talk about stories, really, more than anything else. This is the evidence, and it's published from 2024 for positive selection for the LRRK2 locus based on genetic terms. It's known as the integrated haplotype score. It's looking at the length of a haplotype around the mutation and the variability in it, and looking at it as a ratio of that haplotype versus the other person's haplotype, and counting that across the population. This big peak here is highly significant cluster of elevated integrated haplotype scores, pointing to positive selection around this region of chromosome fold. This is where LRRK2 is in the human genome. We found this gene last year. I want to talk about that, RAB32, because I want to explain the penetrance. RAB32 came from families actually from Tunisia and Canada, and there are a bunch of other ones we found. It also predicates risk. We find these families all over the world. But the mutation comes from the Middle East. This one was originated in the Middle East. Now, I've looked in population databases in Saudi Arabia and in Qatar, and the frequencies that are published in the public domain is around 1 in 3,000 people have a RAB32 serine 71 arginine mutation, which predicates their risk for Parkinson's disease. Not a single patient yet has been reported from the Middle East, from that region, with Parkinson's disease with this variant, which is kind of interesting, kind of surprising. But they're going to have it. It may not manifest as full-blown movement disorder, Parkinson's disease. Maybe it's mood disorder, depression, apathy, constipation, sleep problems, loss of sense of smell, all of these things, the inflammatory phenotype. Maybe it's all wrapped up together, and you'll see that as a constellation, but maybe not the movement disorder yet. But it's there. It's there. It activates low-kinase, and all of these, we found it in all these families. They're all relatives, same haplotype. They're all cousins, no matter whether they're living in Montreal or living in Turkey. Here's a direct example. LRRK2 and RAB32 get together. What's shown here is a picture of a cell that's been infected with salmonella. Salmonella is the big blue thing. And the cell reacts by wrapping it up in a double membrane coat called an autophagosome. That's this thing in yellow. And what it does, it signals, that process signals in part to mitochondria, which are shown here in green. And mitochondria produce a chemical, the itoconate. It's a metabolite of connotates called the TCA cycle for any of the biochemists out there. But if you know anything about biology, you might remember that mitochondria are kind of like ancient bacteria in our own cells. And the TCA cycle, the tricarboxylic acid cycle, is kind of a remnant of that, the powerhouse of the cell. Anyway, TCA cycle makes up itaconate, and itaconate basically it comes from mitochondria and it's injected into the autophagosome. And because it's a bacterial metabolite, in a sense, from mitochondria, it's used to repress the division and the growth of the salmonella in the autophagic vacuole to stop that horrible pathogen from dividing inside the cell. That's the first thing the cell's got to do. The next thing the cell's got to do is got to upregulate its lysosomal biogenesis, increase the number of lysosomes. Lysosomes, for the most part, contain acidic hydrolases. It's a nasty little enzyme that chops things up. It's going to surround the autophagosome with acidic hydrolase with lysosomes, and then it's going to basically infuse the autophagosome with acidic hydrolase to digest this material, to kill the beast. Now this is happening in a macrophage. It's a cell that's drifting around your body. It could also be happening, the same biology could be happening in mast cells, which are involved in allergy responses. Does that sound familiar? Allergy responses? Maybe, I don't know. The same biology probably is going on, and I think it's probably going on, certainly going on in microglia in the brain. So these are the immune cells of the brain. But the same biology, I think, is also going on in some senses, type 2 pneumocytes in the lung. They produce these type of vacuoles. They have different purposes. T cells, same biology going on. Dendritic cell, same biology going on. Dopaminergic neurons, same biology going on, using the same components but in slightly different ways. Now, Parkinson's disease is a multicellular system-wide disorder. Okay, the nigrate degenerates and dies, and we need to know why. But that's not where it starts, right? I think it starts in the periphery, right? And it starts in part with inflammation. But the beautiful thing is, and I want to leave you with some hope, the beautiful thing is that dumb salmonella doesn't have a brain, has no cognitive faculty whatsoever, has managed to evolve its genome to basically create mechanisms called GTGE and SOCD2, if you want technical names. These are effectors that basically the salmonella itself produces that attack CLAB32 protein and either cleave it or inhibit it to enable that salmonella typhi survival to give you a really, really bad dose of gastroenteritis, right? The salmonella has developed a mechanism to be able to survive much better in its host by manipulating its genome and by producing these proteins. Basically, it's managed to overcome. And I think we can take a lesson from this evolutionary example of how we might be able to do the same thing, or we should be able to do the same thing. In part through lactic kinase inhibitors, but in part through agents that mediate RAF32 biology. So that's my last slide, really. It's all roads lead to Rome. It's a map of Rome. There's some really cool papers that have come out. Huge number of really smart people at this meeting. It's a bit intimidating. They're all so well. There's biologists and it's great. And whenever I go, I tell them what I'm working on, right? Because I'm challenged to get funding to do it, and I've only got one pair of hands anyway. But this is it. This is what I'm thinking right now. This is Parkinson's disease. And you're like, what the hell? But it's a molecular and cellular picture of what's going on, of how mitochondria and lysosomes and metabolism, mTOR, is a master regulator of metabolism, get it together with endolysosomes and how they regulate and integrate this mitochondrial metabolic lysosome biogenesis kind of pathway with transcription going on in the nucleus to create these components in the first place. This is a well-honed machine, right, that becomes really stimulated by, you can see interferon gamma up there and TLR4, by these immune inflammatory activators that come down and they signal to TFEB or TFE3 to basically dephosphorylate it to get it to go to the nucleus. And when it goes to the nucleus, it switches on lysosomal biogenesis, and it does it thinking that perhaps there's an invading pathogen in that cell. There's a variety, It's the basic mechanism in a macrophage, right? That I'm giving you here. It's one cell type. It's not every cell type in the body, and we got a lot of them, and they're all very specialized. So we have to think about this in a much more holistic sense, how these cells are working together, because they're also communicating with each other, producing cytokines and inflammatory agents. So I think this basically cycle is a vicious cycle that once it becomes stimulated chronically, it's like waves on the beach. You can't stop it. You can't turn it off. And lots of things can trigger it. It could be inflammation, it could be stress, it could be trauma. A lot of that sympathetic fight-flight response is triggering it. And if we can intervene and just stop it, slow it down a bit, I think we have a very good chance of stopping and slowing down the disease. I really do. I spent 30 years doing it. It's taken a bloody long time already, hasn't it? But we're nearly there. We're nearly there. The beautiful thing is, it's not conjecture. It's not associations. It's causal relationships that can be tested in a cell, in a mouse model, in a human being with a drug. You know, it's not association. It's very directive. So I think that's me, and I got a little team, one of many. Any questions? I'd love to, yeah, I'm around if I've got time. I'd love to tell you my stories. Got lots of stories. Sorry, but like you must need a split second faster than you. At the back, sorry.
Yeah. Thank you. I don't understand it all, but I'm picking up a few threads. One of them, I really appreciated that Parkinson's is more than simply a movement disorder disease.
Oh yeah.
And yet most of our work focuses on movement.
Most of clinical neurology focuses on
Correct.
They're specialists.
Right.
They're movement disorders specialists.
Movement disorder specialists.
That's what they do.
And so we talk about motor symptoms and non-motor symptoms, but you've helped us to understand that it's physiological, it is cellular and molecular, it is immunology involved. So it's much broader. So frankly, it's hopeful to hear what you're saying.
Look, people are finally waking up to it. Even humble little scientists in their little labs who don't see patients very often. You know, I spend a lot of time talking to patients with, for the most part, and families with the most part early onset Parkinson's disease, and that has been so instructive. Still, and it's such a driver, you know, such a driver. I've learned so much about the disease, and so you guys, if you have Parkinson's, I mean, you've got to get the message out. It's worked with me. But medical textbooks, you know, movement disorders neurology, they're specialists. They just do movement disorders neurology. They don't do the rest of it. And the healthcare system's so fragmented. They don't join the dots, you know. But it's far, far, far more than that. As anyone with Parkinson's disease, I think, realizes, you know.
This is hopeful. This is encouraging to know that there are other avenues that may bring not just symptomatic relief, but actually begin to address the origins and functions of the disease.
It's the best way to bring symptomatic relief is to halt the disease course, to stop that vicious cycle from continuing. Because what happens is over time, you end up getting a chronic problem. Maybe it's alpha-synuclein upregulation in the brain, or tau, neurofibrillary tangles in the brain. You end up getting localized inflammation that's basically exacerbating the problem, right? It's like a runaway train. Yeah. Yeah, so I'm glad you got something out of it.
I guess some threats.
So
going back to one of your first slides, where is the evolutionary advantage?
survival. So for example, we put the same mutation, LRRK2G2019S, into mice. And if you give the mice pretty much lethal doses of pathogens, whether it's viruses, It's been done with viruses. It's also been done with Clostridium rodentium, which is like the commonest pathogen for a mouse. The mice that are wild type, normal, they die within a couple of weeks of these infections. The mice with G2019S, they survive. They survive longer. They survive longer because their system is already primed to respond to that to that insult, that pathogen. It's already primed. It's ready to go because their lactokinase activity is ready to go. The doorway is already ajar. Kinase is already active. So that's and we can see the same advantage for RAB32. We see the same advantage actually for pink 1 and Parkinson. Not quite as clearly. The diseases are far more rare and they're recessive. These dominant causes is far more common. It's easier to get the numbers to be able to see this, but it's a survival advantage.
Short term.
Unfortunately,
It's a double-edged sword. It's like yeah, it's a survival advantage to reproductive years, to have kids, whatever. But the double edge of the sword is that Parkinson's disease potentially as a consequence. Right?